programs of the CSIR when he was there 4 5 years back in Delhi headquarters. He was one of the program coordinator for small synthesis of small molecules, and he was involved in the uh, liaison between the institutions, various pharmaceutical research institutions, universities, and the CSIR. So the compounds, I would also request Dr. Haridas Rode to, in short, if that program is still working, then he can emphasize in that his lecture, and even guide the young faculty members who are present over here, how they can increase their research capabilities by making tie-ups with the CSR labs or small projects if are presently available with the CSIR, and how they can go ahead with that. Because nowadays we are seeing that CSIR is playing a very active role in the research field. They are giving research interns in this period also. Some of my students also have joined as a research interns in various CSIR labs. So that is wonderful thing. So I welcome Dr. Haridas Rode. Please can start your experience as well as guidance to all the faculty members as well as research scholars joined over here. Thank you.
Thank you very much, Professor Sharad Wakade. Also would like to thank Professor Pathak, Director DIPSAR, for inviting me to give a lecture actually. And also would like to thank the Professor Goyal and Mrs. Parle Madam for inviting me and give some thoughts on the drug likeness topic actually, which is one of the important topic in the medicinal chemistry. So today I would like to describe two things. One is the Lipinski's rule of 5, and which was then subsequently was developed further by other eminent scientists, and then the rule of 3 came in picture. So thank you all, and I will now straightway move to the presentation.
Please.
Okay, so actually basically I selected this topic. The reason being is that this is one of the important milestone which was achieved in medicinal chemistry in 1990s as far as the drug discovery is concerned. And basically this talk actually is made from the review articles which were published by Professor Lipinski's, as you can see then in 1997 and 2001, which I have referred it, which have this article have given us the rule of 5 or drug likeness, this particular important thing in the medicinal chemistry. So just as introductory part, this slide actually indicates that what is a drug design, and drug development and the whole cycle of the drug discovery and development. You can see that it starts with selecting a particular disease wherein, you want to select a particular target, for example, a protein of interest and so on. Then one need to select the respective bioassay to screen the various compounds against these particular proteins, which is then followed by hit to lead discovery, wherein once the one runs the assay, one will find the primary hit, which is active against that particular protein, for example. And then the next phase of the drug discovery start, wherein hit to lead optimization comes in picture. In the later stage is lead characterization, wherein the large amount of relationship comes in picture, wherein one understand the pharmacophore, that's the features of the molecule which are needed to elucidate the desired pharmacological activity at protein level. And also one understand the toxicophore, that's what are the elements in the structures which are really responsible for giving toxicity that particular molecule, for example. And then you have the lead optimization cascade, wherein activity, toxicity, and pharmacokinetics are further fine-tuned in order to get the lead candidate, which is then mapped for metabolism. That's the wherein the large amount of study carried out in the metabolism aspect to understand what are the metabolites and whether they are active or inactive and what is their in general. Then, of course, the toxicity is mapped in animal models, and also the metabolism is mapped in animal model. And then once the candidate is ready, actually it is evaluated for the possible manufacturing process wherein the chemistry comes in picture, so that one needs to have as pure as the compound need to be, and then at the same time it should be economical feasible. And of course you have then, once it is available, then it goes to the clinical trials where 1, 2, 3 and phase 4 clinical trial, and after phase 3 clinical trial it goes in market. So you can see that it constitutes really a large number of steps involved in that one, in drug design and development, and it's really a costly affair, you can say. And of course you also, at the other side, you always patent at various places the particular drug molecules or the particular derivatives of the compounds of interest. So the slides indicate that it's really costly affair at one side. At the same time, it also takes a large amount of time. You can say generally 8 to 12 years it takes when the starting from the beginning to have a drug in the market. So it's very important, actually each and every step is really dealt with good efforts, and the efforts should be backed with logic. Now, the next slide, where I try to put the ADME, that is absorption, distribution, metabolism, and urination. These are actually the pharmacokinetics parameters. Everyone who studied the medicinal chemistry knows it, actually, but just as an introductory part, I kept it here. At the left side, what you see is that various barriers which drug need to pass. For example, if it is oral, or then you have the GI membrane, then you have the other routes of administration: intravenous, nasal, sublingual, intrathecal, inhalation, and so on. Once it goes to the blood, then you have the distribution, then you have the metabolism followed, which mainly happens in the liver, and finally it is excreted through the kidney. So this is all actually, the activity which happens actually in the body with the drug. So what is important here is that you can observe what are the barriers which are available, for the drugs to be getting into the body. That's also one of the important highlights of this slide. So while we are talking about this ADME, what is important is that you might have understood that it's the oral aspect, that the drug being the oral, actually. So on this, we try to depict, actually what you can see is that various drug categories which have been shown. For example, the drugs which act on the proteases, for example, they have this protease space, you have the kinase space, you also have the lipophilic GPCR space, and so on. But it also indicates that there is a large amount of the chemical space which is available, which is shown in the white block, which indicate that it's oral drug-like space and which need to be explored, indicating that there is a large possibility to develop the molecules in this oral drug space. And this is actually indicates that oral drugs are important and can be further explored for the new drug development. So in early 90s, actually, the scientists were puzzled, actually, by so what they did is that they collected all the data available in clinical trials, and then tried to analyze the molecules which were failed at one side, and the other which were succeeded. So that the observations actually can be seen in this picture, wherein you can see that the drug molecules which failed, so almost 56% were failed in various clinical trials because of the issue associated with the pharmacokinetics of the particular molecules. Or you also have the commercial reasons and adverse effect and toxicity, which also contribute quite well. So this indicates that the pharmacokinetics is one of the important parameters, and this is actually where then the discussion started in the medicinal chemistry field all over the world, in the industry. So at the beginning, that can we understand what are these parameters which we can at the beginning keep in mind, and then which either, if these are required, we should try to put in place, or if these are not required, then how to take away these not useful so the parameters. So can we define such sort of the rules or some sort of applications for any drugs molecule, which will tell us beforehand that whether it is going to be pass through all these clinical trials, or it is going to be good orally absorbed drugs. So that was actually the basic questions which medicinal chemists around the world were puzzled, and that's where actually the lot of activity started. So this or this exercise has happened in Pfizer, where Professor Lipinski was heading the medicinal chemistry group of the Pfizer, and then they were thinking actually whether can we consolidate our all the data and try to figure out what are the factors or what are the observations which can help us in in predetermine whether this particular substance which we are looking at is going to be orally active or not, because pharmacokinetics is going to be one of the factor wherein which contributed most of the drugs failed in 1990s. So actually, this is they come up with the whole studies, and that actually was published in this particular review article, which I am now sharing with you, and where they focused mostly the absorption part. That's the first part, or the first barrier where the drug crosses in. So just to go back, little introduction, as you can say that in 1970s, what we had is empirically based screening programs, where is the drugs were given, to the patient and try to see whether they recover, and that is how actually, so early, the discovery or the early phase of this health system used to work. After that, in 1970s and 1980s, the knowledge base grew that the people understand. They made a lot of observations, and then the observations were supported by the scientific, the inputs, and that's how actually the knowledge base grew. And after that, what came is a phase that's a high throughput screening, wherein in 1990s or 91, where a large number possibility to there was possibility to screen large number of compounds, say 1,000 of the compounds were possible, and that was made possible because of this high throughput screening platform which was developed in 1980s or 1990s and so on. So at the same time, when so in the biology you have the high throughput screening platform developed, at the parallel side in the chemistry or in this chemistry, what you had is that the combinatorial chemistry which was developed, where is a large number of derivatives can or were prepared in a shorter time, and which really complemented both of these things. That is the drug, so high throughput screening activity, at the same time making the large number
Vikas,
of compounds in the laboratories.
I think we lost the contact. We are not audible. You are not audible.
Now, am I audible? Am I audible?
Dr. Dipti, are you able to listen to him?
Yes, sir, I'm audible. It's audible.
Hello, am I audible?
Yes, sir.
Yes, sir, you are audible.
You are audible.
Okay, then I'll proceed, right? Okay, then as I told you, there are two parallel things. That's the high throughput screening in from the biology section, and then you have the this parallel synthesis from the medchem or the chemistry, so which are followed. And that what has happened is that the large number of compounds were made actually, and thereafter you have the phase. So after 2000, you have large efforts actually put in this molecular genetics to understand the various decisions so on. So actually, basically, what happens in 1990s, wherein when you have these two big platform coming together, the large number of compounds were identified as identified as inhibitors. So now you can say in the case of the high throughput screening assay, earlier what is important is that the compound's activity should be measured as possible as in the aqueous medium. But what happened in that case, when you have large number of compounds available at the same time, the screening facility was available, and hence DMSO came in picture as it is one of the good solvents. So most of the compounds soluble in that one. So that is where actually the scientific community started using DMSO in a small amount of DMSO in the assay system, or they also started use of non-ionic surfactant to solubilize the molecules, and then so that the assay can be run and then results can be obtained. So now it is very important observations, as now we are looking in the current scenario, but at that time actually the people were more interested in getting more number of hits, more number of compounds, and so on and so forth. But what happened, having DMS in the screen, now the most of the insoluble compound which are were there, actually they also were tested because of the DMS, so they were soluble. So as a result, what happened is that actually you get inhibitor activity, then it was detected in most of the compound actually, but which had a very poor solubility. That's where the problem comes. So actually the physiochemical profile of that particular hits or the compound which were under progress, now it was no longer was depends on the solubility of that particular compound in water, for in vitro activity, but it was depends on the various other factors, like for example, the medicinal chemistry principle, where the medicinal chemist is more important or more happy if the compound gives activity. That's where it is coming. So he tried to put more bulky groups into that particular molecules of interest. Or you also have the nature of high throughput screening, as I told you, DMS came in picture because one need to screen as many as compounds, and that's why actually. And then at the end, we also have the decision making to select heat for further SAR study. So all these activities actually ended up in selecting large number of compound, but they were deviating far away from their solubility with respect to the aqueous media when it concerned. So what happened actually there? The people or the scientists were able to generate a large number of hits, but they were more lipophilic in nature because of all these requirement which put forward by the use of medicinal chemist activity or towards that direction, or using of the high throughput screening assay, or the parallel synthesis and so on. So this is where actually, this analysis tells that actually we were deviating away from the solubility, that particular parameters of the compounds. And there was then they realized that there is a need actually to restrict, how to filter this compound, because if you select a compound which is less soluble in water, then it is most likely it is going to fail down the line of the drug discovery path, because it is not fulfilling the primary criteria that if the drug has to be orally active, it has to pass to the oral barrier, actually, that one. And hence actually there's was a lot of discussions, lot of studies were going on, and that's where actually the Professor Lipinski's Professor Lipinski and his team actually they did a detailed studies on this one, and they come up with this so-called Lipinski's rule of five. So as you say that why this ADME prediction is important, or let's say absorption is important at the first place. So before going to in vitro or before going to animals and so on, it is important. It's important because it, first of all, as you said, that there is large number of activity involved in the drug discovery and development, wherein one could if one knows that this is going to fail down the line, that's and if we still pursue that one, then we are going to lose large number of resources, right? Then also it also helps to avoid the unnecessary clinical trials if you can pinpoint at the beginning that okay, this is not going to be good drug, or it's not going to be orally absorbed, right? And as in industry, it's often said that the latter a drug has to be drawn, the more expensive it gets, as So it is important that if that particular candidate fail early, fail fast, and fail cheap, so that we can save the resources, one thing. Secondly, we also can save a large amount of money which is needed for this one. And that's how actually I just try to put, if we have so many of, starting with the high, actually, then you get one candidate after so many the various activities and so on. So it is very important that when one need to save the resources, at the same time one need to save the money which is involved in that one. So the Lipinski's team actually, what they started this exercise, and then they tried to see what are the parameters which we can understand, from the drugs which are already approved, for example, and the drugs which are in the clinical trials, and which are, for example, failed in various phase 2 clinical trials. So what they did is that they selected some compounds. These compounds were from the phase 2 clinical data, actually. And at that time, like what we have now, various drug banks wherein, or various the sources where the compound libraries are available, or the drug libraries are available. So they were so at that time what was available was the this word drug index. That's one of the database. Another was USAN, that is United States adopted name, or you have international non-proliferative name. That's another database. And they collected all the phase 2 clinical trial candidates, actually. So if you move forward, what you see is that they started this exercise by taking around 50,000 compound from this World Drug Index database, actually. And then they also compared that database with USAN and INN database, and then they try to figure out which are common in all these so at least in the two databases, and then they figure out that around 3,000 candidates were there, and then they use one more filter to take out the peptides and this poly and cot, this kind of words, or your this phosphorus containing these compounds of the candidates, and which has ended up or they have ended up with 2,245 compounds or the molecules which were in the phase 2 clinical trials, and then they started analyzing this. So what they did is that, okay, they looked at the molecular weight, and what they found is that so almost 11% phase 2 compounds which they considered were having more than the molecular weight more than 500 daltons. In other words, most of the compound which were there available in the database, they had the molecular weight less than the 500. So that was the one observation they could get. The second, they looked at the lipophilicity, which you all of you know that is the measure of the potassium coefficient in in octanol and water. So they used actually this the partition coefficient and try to figure out what are the values. They converted this log 10 to the base of log 10, so that the values are easy to understand or relatively, because as the log phi, these values are very close by, so to convert them into relatively distinguished way they use a log 10 anyway. So they use a various programs. So one is like, for example, this Pomona College medical chemistry program was this available, this computational program, or there's another way like mlog-p, which was described by the Moribuchi. So these are the different methods or the softwares which were available to calculate, theoretically the log-p value. And what they found is that almost 10% of the compound had more than 5 C log-p. In other words, nearly 90% of the compounds which they were analyzing having C log-p which was less than 5. So this was the second observation they came across. Now what happened, they also looked into the hydrogen bond acceptors and hydrogen bond donors, and what they figure out from the data is that, or in general, actually what you can do is that you can just add, the NH and OH from the compound which are looking, and then that should be give a rough estimation of donor actually. So in this case, they found that the hydrogen bond donor, almost 8% of this compound in the particular library were having more than 5, hydrogen bond donor and X. So that was another important observation. And we also have the hydrogen bond acceptors. When they looked or they summed all N's and O's which are available in the molecules, and then they found that only 12% have more than 10 N's and O's. In other words, almost 90 or 88% of the compound which they have less than 10 value for the hydrogen bond acceptor. And this has actually, when they sum up, when they put all these observations together, what they could know is that the poor absorption of compound is more likely if the compound has compound has molecular weight more than 500, its log-p is over 5, there are more than 5 hydrogen bond donors and acceptors, and there are more than 10 hydrogen bond acceptors. So actually this has some of the rule or these observations in the form of rule of 5, which gives an idea the likelihood of that compound to be orally absorbed. And then this rule was further extended to include the polar surface area if it is the compound having polar surface area equal to or less than 140 angstrom, and double bonds equal to and more than 10, where the two more parameters were added into that one. So this way, actually, in 1990s, they were able to understand theoretically that what are these physicochemical parameters which has a large influence on the availability of a particular compound in question. So here you can see that actually during the time they put this GlaxoSmithKline library, and then you can see the molecular weight and the log-p, and then you can very easily see that the compound which are low, the C log-p is lower than this 5, for example, and then the molecular weight is lower than 5. And this is where the quadrant actually, which tells that the likelihood of this compound to be orally absorbed. So Okay, well, this rule of five was designed, and then this was a good guiding tool for the medicinal chemists to use, to decide whether this compound is going to be orally absorbed or not. But of course, there are some, outliers actually in the rule of five. So, for example, the compound classes, these are substrate for biological transport, because all the rule of five applies to, the passive transfer, the substances of the drugs which are, transported into the body passively. But you also have the substances, which are the substrate for the various biological transporter. For example, you have antibiotics or antifungal, you have fungicides, you have vitamins and the cardiac glycosides, which do not come under the rule of five. So you also have the limitations. What you have is that it applies, of course, to only the oral compounds. Secondly, it applies to the compounds that are absorbed by passive transfer, as I told, and there are exceptions, as you know that. But it also tells that, so without doing any experiment, tells that the compound is, either a good or a bad oral drug. So these are also some of the limitations one need to take into account, while going, while designing these experiments or while designing the compound library for the further drug discovery. So this is what actually, they did analysis of the novel chemical entities which were introduced in 1990s to 1993, where I have given the source actually where the reference is there. They found is that, that 133 novel chemical entities actually, um, they have molecular weight less than, or I mean, around 408, up to 1.8. You have hydrogen bond donor around 2.5 and hydrogen bond acceptor 6.9, indicating that it is well fitting into the Lipinski's rule of five or Lipinski's these observations what they come up with, of course. So as I told that, when they framed the rule, they also developed a software for their this pharmaceutical company, and nowadays each pharma company has their refined software wherein Lipinski's is the rule of five. Then it was the rule is further tuned down to adding some more parameters and tuning down these parameters so that they use this as a filter to guide their drug discovery process. So here actually I just highlighted few of the example wherein you can see that the underlying part, it says that okay, this is a particular drug, but it really violates. For example, here in alert you can say that there is one alert indicating that this NNO, that is the hydrogen bond acceptor, this is now this particular parameter is more than what one would have expected, and so forth. You can see in other examples. So in the summary of the Lipinski's rule, what we have studied is that actually when the pharma industry was going more to the synthesis and high throughput screening, but it is also important that one need to stick to the as close as possible to the experiment in the animals or experiment in the humans. So wherein we have the aqueous as a medium for the experimental purpose, and of course it is still elusive task. Still we have these guidelines or tools to decide certain parameters, but definitely as 100% proof that we still need to look for, or we are still looking for. So what lessons we learned from this Lipinski's team's activity is that okay, we have the Lipinski rule of five, and we have now this physicochemical parameter as the guiding path to take the molecules forward. But at the same time, we also need to look for the reactive or unstable group, which if they are present, you have to really take into account and then take out that compound from progressing forward. You also need to have the diversity of the compounds to have more likelihood of getting the new drugs actually. And now, so in the recent time, actually, there are various softwares which are used to calculate the various parameters like hydrogen bond donor acceptors and CLOPs, and even further the blood-brain barrier transport and the excretion and all other data. So there is recently, in few years back, I think the another software which came in picture, that's a Swiss ADME. So it is a free software actually, and one could use to calculate the various parameters, as I said earlier, in addition to few more parameters. So this is a free software, and one can use it for their academic purpose, or if they are doing some research that can be helpful to generate some data and support their their molecules and so on. So now I'm just changing a gears little. What I have explained till now is the Lipinski's rule of five, wherein which will gives the possibility, for a molecule which is going to be likely to be orally absorbed. But now I'm just moving to the rule of three. So this rule of three, now this is a little different topic, wherein you have in the medicinal chemistry you have various strategies. So for example, the screening strategy wherein the compounds are screened against a particular target. We also have wholesale screening activities, and we also have the fragment-based drug discovery approach. So I'll just give 1-2 slides, sort of introduction. So here you can see that this is, for example, ribbon structure of this kinase, where you have the N lobe, we also have the C lobe, and then which is connected with the hinge region. And this is actually place where the substrate sit, that is ATP sits, and then this kind carries out the phosphorylation of the other proteins as well. So this is one of the important target which is overexpressed, in the cancers and so on. So these slides actually, this indicates that, for example, this is one of the drug that erlotinib, which binds to the active site of that kinase, wherein you can see the hydrogen bond happening between nitrogen of this quinazolin, for example, here with this methionine and threonine amino acids and so on. And this also, we can see that hydrophobic pocket which is occupied. So this indicates that this is a slide I just wanted to show, the interactions of the inhibitors with the proteins and so on, because when further we'll go to understand, what are the fragments and how the fragment library can be designed, this will help in. So moving forward, actually, again, we also have the chirality. That's wherein, the various substitution on molecules and how they can fit into the various packets of the receptors also is very important, actually. So this arrangement of these smaller fragments, on a big scaffold is also very important. So as I mean, this is one of relatively the recent, in the sense in 2000 or so, this fragment-based drug approach started, and you can see that in the high throughput screening, in the first case, what you have is that you have the all the compound which are made by the classical medicinal chemistry, based synthesis, or you also have the parallel synthesis. So you have the compound, you just put into the test tubes and then try to see whether they binds to the particular protein, and so this is what you get a picture. Now in the case of the fragment, you have much more smaller, they are much more smaller in nature. We can say that, for example, here in green color, you also have the another fragment just sitting to, in the blue color and occupying the next pocket of this enzyme. So one can then bridge these two, and then one would get a better fragment, and then this can be grown. You can see another fragment which is binding. So this can be tethered together or bridged together, and then one would get a better inhibitor. So this is actually what the approach is, wherein you start with the small fragments and then you grow that fragment with respect to the activities concerned. So this also can be seen from this article, actually, which was published in 2005. So what we have is, two approaches. That is the the classical medchem based approach, and we also have fragment based approach. Now, when you compare these two, the hit rates, in the case of the fragment based approach, they are really relatively high. And here, actually, you try to pick the actives. When you say that, okay, this particular fragment is active, then it can be in micromolar, or in sometime it can be in millimolar. So this affinity is very low, but it can be picked up very fast, actually. And then there are more chances of growing that particular fragment into the drugs, which is like drug like this compound, because now we have only the small fragment, and there are ample amount of opportunities available to grow that forward with respect to activity by adding the various groups and so on. So what you have is that you have the smaller size. It is a simple one, actually, and it is better soluble. So it can be a good starting point for this drug discovery approach. So what you can see that the important aspects in the drug, this fragment-based drug design, are when you set out to look for the library, which library is very important for this, the fragment-based approach. Then you have the three components. One is the library design itself. Second, the screening methods, what is available with you, and then of course the medicinal chemistry expertise which is available with this. Which all three, sums up to give good design for the library. So in the case of the library design, what one need to look for is that the smaller the fragment, it is better, so that you have the less complex. And if you start with some sort of privileged structures in a small molecular data cycles, that also is a good idea. And as far as the screening is concerned, once you have designed the small set of these fragments, and then one could go for the NMR based screening or X-ray based screening, or you have the biophysical method like mass spectroscopy and so on. And of course, you also back up with all your efforts with the classical medicinal chemistry based effort. So, but so at the same time, it also need to look into that the fragments of high solubility, you know, if you are saying that you want to observe the hits or the fragment which is showing activity, but at millimolar level or at higher micromolar level, then that solubility can be a problem, because if the solubility is low, then that is an issue. You also have at the higher concentration in millimolar, then this particular fragment may tends to aggregates, and then that may be non-selective inhibitions what one would observe in that case. So there are around more than 26 protein target actually were have been targeted, by this fragment-based drug discovery approach. So these are here are a few of the examples actually where you can see that in the first case that A, B, C, and D. So they have used a very so most frequently used approach, that is initially you identify a fragment and then you add a group so that it becomes more active. So you can see that in the red color here it's shown that this particular, and then it was grown forward by adding few of the groups. Then again, you also this in the case of the B is the same case of the C and the D, that the initially identified fragment they were further grown by using the chemical modification, as you can see. You also have the second, that's the E, wherein two fragments bind at the two different pockets in the enzymes. For example, yellow color, and here is a red one, and then they can be reached together to make the more potent inhibitors. Again, the same in the case of the F here. So the two fragments binding at two different places, and they are reached kind of with this phenyl ring. But here, so in the case of the F, this is called as specialized techniques, wherein you put or the group which can then be reactive with the protein. For example, here is aldehyde, where a nucleophile from the protein side can come and can attack and can form the reversible covalent band. So this is also called as extended tethering, or this is the special techniques wherein some reactive groups are added into the design. And now we also have G and H, where is in situ coupling of the fragment is performed. That means we have the one fragment, another fragment, and then they are incubated with the proteins, and then they react each other, and then they form the covalent bond, and then this then become as inhibitors, which really targets that particular protein. So these are the various strategies actually which are used in this fragment-based drug discovery. So this is slide actually which indicates how the progress has happened. You started with this red color fragment, and with the chemical manipulations you ended with this one. So you started with EC50, that's effective concentration as 50%, around 300 nanomolar, and then finally ended up with 1.3 nanomolar. Against this another story wherein you started with this fragment and ended with Ki of 0.01 nanomolar, wherein the starting is 122 nanomolar, and the some more examples. Also, you have some protein families like these proteases, where they recognize small chemical entities actually. For example, hydroxyethylene, these units generally, which is now shown here in this red color, which is recognized by them. So building the library based on that small fragment has then resulted into the large number of drugs that you can see there, sacubitril, lopinavir, and some other these anti-HIV drugs. So these are some of the successful example wherein we have the lead, as you can see at the left side, that is angiotensin 2 receptor antagonist, and then further manipulation with respect to that so virus activity. Then we have this losartan, which can as a, also have, and this AC inhibitor by by taking this fragment and the chemical modification, it ended up into the generation of this captopril. You also have this receptor antagonist started from here, and then you have the phase 3 clinical trial. It was that is Untitled and so on. So these are the few examples which demonstrate how the fragment was taken as a starting point for the drug discovery and then ended up into the successful drug molecules. So this activity has been summed up in the form called as a rule of three. So this rule of three is for designing the fragment for the fragment drug discovery. So wherein one can see that the compound or the fragment which has generally molecular weight less than 300 Dalton, C log P is less than or equal to 3, and then hydrogen bond donor less than 3, hydrogen acceptor less than or equal to 3. So these are the important these physicochemical parameters one need to put, before you design the fragment vector discovery library. And in addition, it can be extended by addition of number of rotatable bonds and then polar surface area, as can be seen here. So with this, actually, it brings to the conclusion of my talks. I would like to thank the organizer for giving me an opportunity to discuss, this drug likeness and its implication, in drug discoveries. And also would like to thank all the participants for participating in this. Over to you, Professor Wakodi. Thank you.
Thank you, Dr. Haridas. It's very nice and lucid presentation you have given for this QIP delegates as well as PhD scholars who are listening and watching this presentation on the Zoom meeting also as well as on YouTube live also. They are seeing it. Now the session is open for the question answers or any discussion if you delegates wants.
Hello.
Yes.
Yes, yes. Dr. Gajan, please.
Hi, yes. Good morning, sir. And Dr. Haridas, thank you for a nice presentation. Sir, I want just a question. We have a summary for Lipinski rule of 5 for organic compounds. Sir, can you give me some idea about for protein structures like that?
No,
Summary for protein structures.
I think summary for protein structures will be difficult. The reason being is that when you have the small molecule, you have possibility to control various, aspects which you want to test. But protein being a large molecular weight compounds, it is going to be very difficult. And second important thing is that the proteins, they are the parts of the biological system. So they are one cannot consider them as an isolated system, because they are the part and they also talks to the other proteins, and it's very complex in that way. But what Lipinski's rule of 5 try to give some guidelines to the medicinal chemists. But as we move from 2000, there are large number of additions has happened in the Lipinski's rule of 5, and each of pharma company they have their own rule of Lipinski's. I mean their own set of rules which they use as a filters, which are not available to the public domain, of course. But each pharma they have their own rules. So this has given the good emphasis, or good knowledge that what one would use to design actually pre-select. But as you said, for the protein it is going to be very tough and it will be very complex.
Thank you, sir.
Thank you.
Really a good talk. Even I would request Dr. Rode to give some highlights or type of work you people are carrying out in the IICT, Hyderabad, type of
Yeah.
projects they are doing, or any type of work in which the MPharm students or PhD students can enter that lab, especially any collaborative type of work, if it is possible also.
Sir, your voice is not very clear, Wakode sir.
Yeah. I request.
I got it actually. I got it. It's a little low, but I got it. Yeah. I think we in IICT, yes, in the medicinal chemistry and in allied area, we do work in various platforms like this cancer, anti-inflammation, tuberculosis, and neurological disorder. So wherein we also have the synthetic chemistry or the medicinal chemistry team, as well as the biological screening platforms are also available. So one thing is that actually what IICT is having is a national mole bank, which where the large number of compounds can be stored. So therein actually, if you would like to screen the compounds, various the platform which, for example, are available with you, then that PI can contact to our mole bank, and then that compound can be sent it to the mole bank. They will be preserved, under inert atmosphere, and then whenever the screen comes, then that can be used actually for the screening. And then whatever data is generated, then that will be shared, so with the PIs as so as a collaborative work actually under the MOU.
Hello.
Yeah.
Sir, I want to one more question. That is there any facility that you train the faculties at your institute or at your center?
I think there is the one DST has one program actually, the visiting faculty program, wherein they allow for I think 3 months or so, wherein the professor or a teacher from the other academic institute can go to various CSR lab, or it is to other national labs as well, and they can do some research for 3 months and then train themselves. So there is a special program by DST actually on this one.
Thank you, sir.
Actually, PhD students or the faculty members from small organizations or institutes would like to screen their compounds or especially spectral analysis. That type of facility is available with the IICT, which is available in NIPER or CDRI, but they take some more time. So, is there any such type of facility available there?
Facilities are there actually, but there I think some small amount of money probably is needed to get this done.
Chargeable basis also, no problem.
And of course, one also need to understand the load and all these things, because it's a first come,
That
first
is
served
the
basis. So it is the cycle actually how it goes. Yeah.
Any
Hello.
other? Please.
Hello. Sir, this is Sandesh Lodha from Maliba Pharmacy College, Gujarat.
Okay.
Sir, is it so stringent to follow Lipinski's rule of 5 whenever we are doing for some computational studies? Because there are many molecules which does not follow Lipinski's rule of 5, but still they are good enough.
Yeah.
What is your take on that? Because natural compounds, they may have a complete different perspective to look at.
Right.
Dr. Sandesh, can you put on your video, please?
Network issues was there. I think yesterday we tried, but
Yeah, I tried. There is some network issues. Still, I'll try again.
It's okay. Okay.
Is
So
it visible?
okay.
Yeah, it is visible.
Yeah. So your question is okay. So it's a really very good question. But as I covered in my talk, that there are also limitations, and so it is not 100 foolproof. Say that okay, you follow and then you are successful, right? There are still but this what it gives, and the general feeling, the general idea, whether it is likely to be orally absorbed or not. Yeah, of course, if it is from natural, sources, marine sources, then it is most likely to be the substrate of your transport and so on. So I think in that case, you do not really need to worry. But if you are, handling, let's say, for example, thousands of compound, you have a library of thousands of compound, but you don't have resources to carry out screening. You probably have, the possibility to screen only 100 compounds. Now, how do we do? Um, so in
Yeah.
that case, actually, you apply some sort of filter, which will give some sort of guidelines. So it is the guiding mechanism actually. But one cannot say that all compounds will fall in that one. So those who are, for example, now in this case, then one can do the in vitro and in vivo this probability study, and then try to figure out, okay, if it is okay, then you proceed further.
Okay. I am an alumni of CPL, and I was with Ramdas for 2
Yeah,
years.
good to know you. Yeah,
Ramdas Rode. Yeah.
yeah, good to know you.
Yeah.
My brother, yes.
Hello.
Yeah, yeah.
Should we start?
Thank you, sir.
Should we start?
Madam, yes, madam.
32 students have joined.
Any other queries? Any other queries are there? There is one query, sir.
Yeah.
The query is, please suggest more open softwares for ADME predictions.
Sure. I think I'll go back and I will share with Dr. Wakore so that he can circulate, the whole list of these
So nice.
these softwares.
Then there is one more. Can IICT help DIPSAR scholars for biological activity?
Yeah, as I said that, I mean, there is a possibility that, I mean, so rather scholar, their guide will come in picture, so they can collaborate with, so with the scientists, and then it can be taken forward. Yeah.
Right, right. Wonderful presentation, Dr. Haridas. We are grateful to you for sparing your time and contributing significantly to the knowledge of the student, knowledge of the delegates and our PG PhD students, especially about the Lipinski's rule of 5. And it was a very well presented lecture. And let me present you with a certificate.
Thank you.
Kindly receive the certificate. We will be mailing you the certificate. There is a flower of a bouquet of flower as well for you. Hope you like them. It's a combination of nice white and purple colored orchids.
Oh, okay, okay, very good.
So thank you so much for sparing your time
Okay.
and giving a wonderful lecture.
Thank you, madam, for organizing this talk, and also thank Professor Sharad Wakode for doing all the communications and organizing this one, and of course Professor Patel, director, is for really thankful for giving us present this platform. Thank you very much.
Yes, thank you very much. It was
Thank you.
nice to you and interacting.
Yeah, I will leave now.
Yeah, even our students would like to be in contact
Just a minute.
with you. Yeah, Dr. Madhu, you would like to talk?
Dr. Madhu is there. Kindly, I request everybody to mute their microphone.
So, thank you, Dr. Haridas.
Okay, thank you. Yeah.
Thank you for your valuable time and guiding all of us. Thank you.
Thank you. Thank you. Thank you very much. Thank you.
Wakode sir, our next speaker is ready.
Yeah, within 5 minutes we'll be there, but we can start the introduction of few delegates because we are lagging behind. 4 5 delegates we can cover today also. So that
Sure. Why not, sir? Is Shrikant Joshi there? Dr. Shrikant Joshi?
can,
Yes, ma'am.
switch on your,
Can you introduce yourself, please?
Yes, I'm
Please
Dr.
put
Shri.
on your video.
even all of you can start your video also for the time being, so that we can see the actual photographs, or sometimes photographs are also not there, from which organization you people are there, the details of that organization, where from you have done your PG, PhD, within 2 minutes only, because next speaker is
Yeah.
about to join. Yeah, he's joining.
Yes, sir. Sir, I am Shrikant Joshi. I have completed my MPharm in pharmacology and PhD in Veer Narmad, South Gujarat University, Surat.
Okay.
I am working as associate professor in Maliba Pharmacy College, situated at Bardoli in Surat. My areas include and as well identification of various targets and use of in silico techniques for drug development.
Great to know you. There are 3 participants which I am seeing from Maliba College of Pharmacy. Gajanan Kalyankar sir,
Actually, 4.
then 4. Great.
Yes.
Maliba is good, doing good research, and we could visit Maliba, say 1.5
Yes, ma'am,
years
always welcome.
back.
Yes, ma'am, always welcome.
Yeah, we visited actually. I request Madhu ma'am to mute her video, please. I request Madhu ma'am to mute her video, please. Madhu ma'am, kindly confirm your muting.
Hello.
Who's this, please?
Madhuri Deshmukh. Hello.
Kindly introduce yourself.
Myself, Mrs. Madhuri Tanajrao Deshmukh from Rajgarh Nanpit College of Pharmacy, Bhor. And my subject is a pharmaceutics, M.Pharm in pharmaceutics.
Good
Hello.
to know you.
Yeah.
Hello.
Yeah, you are audible.
Okay. Thank you, madam.
Thank you very much. Good.
Avneet, can you introduce yourself?
Hello.
Yeah. Dr. Madhuri. Yeah. Madam.
Madhuri, thank you for the introduction.
Madhuri Deshmukh.
No.
Okay. Okay. Thank you, madam.
Avneet, can you introduce yourself?
Next speaker. Avneet, unmute yourself.
Good morning, everyone. A warm regards to VC sir, Pathak sir, Vakode sir, Parle ma'am, and other faculty members. I am Avneet Kaur, ex-student of DIPSAR. At present, I am working as assistant professor in SGT University, Gurgaon. I would like to thank coordinators of QIP for allowing me to join this program. Great efforts from Parle ma'am and Vakode sir for arranging these good lectures. Earlier, I have participated as a volunteer in QIP program, but now I feel privileged to be the part as a delegate of it. Thank you, ma'am, and thank you, sir.
Thank you, Avneet. Thank you. Our next resource person, Professor Sanjay Wagh sir, is also there. I request sir to start your video. Yeah, he is there. And unmute. Yeah, unmute also there. Madam, please go ahead. You are on mute. Madam.
We have with us Professor Sanjay Wagh. Professor Sanjay Wagh has more than 34 years of research experience. He started his career at Lupin Laboratories, and after handling R&D for 11 years in custom synthesis unit, he has produced more than 1,000 custom synthesis products for the export. He has produced more than 5,000 screening compounds for drug discovery research, only export. From 2006 in Japan market for Konica Minolta, he has produced 7 doctorates and 110 M.Pharm candidates. He has developed 6 thermal dyes, novel discoveries of anti-cancer, colonal drug delivery formulations, and burn skin care silver nanoparticle formulations are applied for the patent. He has 25 publications and patents. His 2 patents got published. One is azopolymers and second on silver salts. For last 8 years, he is with Fine Chemicals and API manufacturer at director level. He has visited China 15 times for developing manufacturing facility. He has represented the company at London, Amsterdam, Munich, Tokyo, Frankfurt, Paris, Madrid, Barcelona for CPHI. So he's a very well traveled and very well versed person. He has established an API manufacturing facility right from ECCT CTO level in a record time of 14 months. He is a director, Survival Technologies Private Limited, Ankleshwar, Gujarat. He was director and CEO at Common Pharma, Kudalore, Chennai. Currently, he is director, technical at Wadi Varhe Specialty Chemicals, Mumbai since January 2017 till date. He is director at Rumit Chemisynth, Mumbai from 2018 till date. He is a technical advisor for API and intermediate plant of Sahkar Maharshi Shankar Rao Kolhe Sahkari Sakhar Karkhana, Kopargaon. He has completed many assignments like basic technology development, particularly import substitutions, innovative and cost-effective routes of synthesis, development of a business model suitable for given infrastructure, conversion of solution phase reactions to catalytic processes. He has supervised the entire activity from technology support to business development except operations. He has developed business in Japan, Europe, USA, and raw market. He has developed bulk API and intermediate technologies which are cost effective, eco-friendly, and high throughput. I request Dr. Sanjay Wagh to take the dais and explain me first what is ROW market.
Rest of world market.
Rest
of the world market. Yeah, that except the USA and Europe, we call it as a rest of the world market, which is non-regulatory market.
Right. Wonderful, sir. Wonderful. We are looking forward to listen to you.
Yeah. Thank you, madam. Thank you for a nice introduction. And shall I start now my presentation?
Please.
Yes, sir. You are.
Actually, I'm putting my PowerPoint presentation.
Yes, we can see, but still it is blank, sir.
Yeah. Can you see the presentation slide?
We can see just the blank slide, sir.
Slide here.
Yeah, just a minute.
Yeah, no. So, for all the delegates, I'd like to say that my sincere regards to Professor Wagh sir. So what we are today as a faculty members over here is because of Professor Wagh, who was our teacher, mentor for the organic chemistry subject over there. So Sudhakar, Garat, Pai, Then we have listened to all the other members from the College of Pharmacy Nasik. So we are very fortunate to have Professor Wagh as our teacher, Professor J.S. Shetty as a principal, then Professor V.K. Moria sir, Professor Dhake sir, Professor Yadav, Professor Kasture, Professor Derles, Professor Pingle. All these dignitaries are there in the College of Pharmacy Nasik. We are there with
Sir,
us.
we can see your video, the slide now.
Yeah,
Okay.
now slides are there.
Maximize it.
Yeah, yeah.
And sir has got a very vast experience in the pharmaceutical, especially bulk drug industry. And in our college days also, so many pharmaceutical industries or the bulk drug industries used to contact sir. Even the research topics or projects which sir used to give to the MPharm and PhD students was industry oriented. So I know one of my senior, Dr. Shikhande, he was working on some waste product of the sugar cane. So what is there as a waste, मड़ी जिसे कहते हैं, okay? From that also he used to extract or isolate the chemical constituents which are biological or chemical interests. So sir, please go ahead.
Hello. Good morning, everybody.
Yeah, we are able to see.
Slide change. Adire, slide. Good morning, everybody. Now my talk will be slightly offline of your whatever currently I've seen the previous talk about the discovery methods and the screening libraries and lot of academics. My talk will be more commercial talk. Means looking at the current commercial scenario of the API industry, and as I consider myself as a blend of academics and industry. Because I worked in pharmacy college for more than 29 years, and also the same time I was associated with the industry. So I was simultaneously looking at the academics as well as the industry, and I tried to bring so many projects to the college, as well as we had a big program of for Lupin laboratories for training their staff for almost 3 years. Lupin and the GSK, we have trained the staff. Means I right from the beginning I tried to bring the industry and academia together for a mutual benefit as well as the benefit of the nation. uh, so today, now I'll start with the first slide. Actually, now we can see here that see we are very strong in API and formulation. Very strong, really speaking. Say like the 50% of the global demand for various vaccines we are sharing, means we are producing in India. 40% of generic medicines to US, then 25% medicines to say UK. So you can see now, and even this, particularly the HIV, is very interesting. 80% of the drugs for HIV, tuberculosis, we are offering to the world. And interestingly, I'll tell you that WHO wanted to treat the patients in South Africa, particularly for the HIV. And when they asked for US to give their offer for the treatment cost, so US offered about $1,200 per annum for every patient, per patient, $1,200, $1,200. Against that, our Indian companies like Cipla and one more company, they have offered at just $193 the treatment cost for the whole year for the HIV patient, HIV infected patient. So you can see that we are very competitive. We have a total business of 1,40,000 crores to export and local business of the pharma. And compared to China, I can say that we are very strong in QA, quality assurance, the documentation, the impurity profiling, dossier filing, all this we do very permanently. And our companies are very strong in this field. And they are catering the needs of all the European and the US consumers. We are also very strong in the analytical method development, impurity identification, their characterization, everything, the spectroscopic methods, very strong that way. Then the European and US API market also, we are leading. Formulation industry is very strong in the ROW market. I would say maybe regulatory market, they are handling their own formulations, but the ROW, the rest of the world market means except say you can say except US and Europe, we are very strong entire world. But now where we go wrong? Why? What is the basic problem with us? Is now the current situation of China. You can now everybody knows our API industry is dependent upon 65 to 70% of the key starting material, which is to be imported from China. We do not produce in India. This is a very, very big problem. And several times our Reserve Bank also advised the government that we are building up the imports from China, and we spend a lot of foreign exchange for this import. So I want to highlight this part of I think this is, yeah. So I would say the hurdles. What are the hurdles here and the problem we are facing? So we have KSM. KSM is the key starting material, uh, key starting material required for the API manufacturing. See, for I can say any drug you can take, like the telmisartan. Then the telmisartan, the key intermediate, we will have to import from China. Even you take many, like clotrimazole, the 2-chlorotrioxide we have to import. So, like these are the big problems for us. And another problem is the Chinese facilities. They mostly use the catalytic processes, so we are not very strong in catalytic process in bulk drug industry. Then our volumes of production also low compared to China, so we should have the large volume of production. Then we are very poor in the biotechnology. I can say the fermentations, particularly the antibiotics produced by fermentation. The entire world, the azithromycin. See, we have to import from China. There is no other way. We are not producing. Mupirocin, so many you can say list of antibiotics. The entire world, the 85% of the antibiotic market is captured by China. So what is the reason? How they achieved this? How they have gone to this level, and why we are not able to do this? So this is the I would say I would like to talk about this part. Just yeah. So now we come to the our academia. Now I know that several times I visited China, and I know that how China has come out of this with the wonderful chemistry and the technology. See, I know there are the border issues, and China is not our good friend and all this, but currently I would say that there will be a knowledge war. Means the one who has a strong knowledge, he will be powerful. There will be no physical war in the future. There will be knowledge war. So we have a very big academic say network of the academic institutions all over the India. And if we want to combat with our enemy, I would say, or we combat with the neighbor who is our troubling us, then we should see that how he has come out to this problem, how he has come the world leader. So there, the academic institute's role is very, very large in industry. I would say the conversion of knowledge to wealth, that how the knowledge of a professor is going to the industry and they are producing wonderful antibiotics. And the professors are holding 30% stakes in the industry. So the knowledge should be converted to the wealth, or the knowledge should be used for the building of the nation. That should be the role of the academic institute. So in this respect, there are a few slides which I would to discuss. Now, what we are doing, if you see, really speaking, I'm not criticizing anybody's skills, and I just feel these are my feelings toward the nation building, that we waste our time in the topic, like dissolution rate, dissolution studies, so many things which are not useful, which doesn't give any of the, all industries are doing their own testing and all this. So one should not go for that. So the students, what students should do at the, say, the PG level or the UG level, they should be well versed with the industrial requirement, what the industry needs. When a student passing B5 and going to the industry, then he see a very different scenario. When he writes a thesis, he can write anything, but when he goes to the industry, then there should a real thing. He should not, he cannot write anything. He has to follow some regulatory. He has to follow so many things, calibrations, so many things. But he is not aware of that. He is not listening to the lectures. He is not doing very well. That is what I, that is my, what I strongly feel. So it is the role of the teachers and the industry to bring the industrial culture, I would say. See, industrial culture is highly disciplined, you can say. Means suppose you have been appointed as, say, the officer in the QC, then the, maybe the industry may spend for some time to train you. But then whatever salary they are paying, they will just see that whether the salary I am paying him, whether I am getting that much of work out of it, which is not there in the college, which we cannot see the commercial angle of that. If I'm using one HPLC, how many samples I'm running on this HPLC, the industry will evaluate that this I have invested 20 lakhs, then I must be able to run so many samples, which not done at college. We keep the HPLC as it is kept. There is no value for the capital investment. So this type of topics I would highlight, and the faculty members, I would strongly suggest that they should be involved in the consultancy projects. They should interact with the, whether they are capable or no, they can do many things, I would say. Even it's not all the time, it is only the bench work and all this. There are many areas. The documentation part, if the faculty members are involved in the documentation part, then automatically the students will get that knowledge. The knowledge will percolate to the students. And even the analytical method development, the college can institute can contribute a lot. They can see so many, they can run so many trials, they can make the derivatives, they can, and then they can see the because HPLC, the big problem is that the response factor, maybe the impurity may be lower, but then the response factor is higher, and you get the false result. So this optimization of parallel methods, quantification, a lot of things are there which college can contribute. Then you can invite the industry people, but not just for some topic or some lecture. He should actually share what problems he is facing, what support he needs from academia. It should be very interactive, particularly at the PG level. They can do all these things, and you should not give the topic like conventionally what we are doing for the MPharm and PhD. The topic should be directly related to the industry, like what China is doing right now. And this is quite possible here. Just a minute. Now I'll go to the next slide. Now I'll come to the solutions. These all I discuss all the problems. Now, what are the, how we should resolve this and how we can come out of this problem, say over a period of say next 5 or 10 years, how we can reduce the dependency on China, how we can build up our HR, skilled HR from our own institutes and of the industry. So it starts with basically your syllabus. Now the syllabus designing, the syllabus should be designed only on the basis of the needs of the pharma industry today or coming 25 years. Now, whatever we are importing, we must stress on that we cannot make azithromycin, we cannot make antibiotics, fermentation. So we should have the objective, because for any syllabus designing, there is an objective. What is the objective for the syllabus? So that should be properly defined, and you must involve the policy makers of the government. the statisticians, as well as the renowned professors, renowned industrial people, when you design a syllabus. You should not, we don't want the mechanical workers. We want the real skilled people which will cater the needs of the current needs of the nation. And secondly, the syllabus must be designed by the universities. Why universities? Because universities have got multidisciplinary departments. They have the biochemistry, they have organic chemistry, they have engineering, chemical engineering, so many things. And you know that the interdisciplinary work, lot of interdisciplinary work has given the good output. So with this infrastructure is available only with the university. It is not available with the regulatory agencies. So it should be with the universities, and universities should interact with the industries. Universities should interact with the policymakers of the government, and they should put together their efforts to design the syllabus. Now, when you come to the research work at the PG and PhD level, now I see that even in my college, I was when I almost 30 years I was there. The cognosy people, they will just take some this thing, some plant extract or bark or the leaf. They go on extracting this with the various sort. Why you do all these things? I mean, why we shouldn't look at the different angles? Why don't we look at the commercial angles? So what cognosy people should do actually, I feel that they should see the volatile oils which are commercially used. That is a need. We need the natural oil, the volatile oils, the originally the natural oils. So they should choose the projects which are directly commercialized, and that should be sufficient for the MPharm, I would say. One should not do some research and do some activity and all these things. Why we do that? Because it is all known, and we waste the lot of animals and these things, and then because the research is not at all directed. Similarly, for the sutics, for chemistry, or even ecology, we are doing so many things, and which is not at all the it doesn't give much output of this. Now, the research programs for either the PG or for the PhD, that should be also decided by the competent authority, like CSI is there, DBT is there, DSTs are there. They should design, they should decide the topics, that these are the topics we have we are spending a lot of money on the import. You develop technology for these antibiotics, for these intermediates, and they should distribute to the universities, and they should conduct the programs accordingly. We don't have money to spend just like that for producing non-fruitful results. That is not useful. Or we are repeating the same things, and we are producing so many theses which has got no commercial value. We should do only the applied or the commercial research. That is what I feel, at least for coming 20, 25 years, so that our economy is strengthened. See, like I can say, antibiotics, like we can distribute these topics to the various industries, uh, like those who are strong in the microbiology, fermentation, all this, then they should go for the antibiotic based on fermentation. Now is a big area of chiral catalysis, the asymmetric catalysis. Most, you can see now all the drugs which are chiral, always there are two isomers when you produce synthetically. Always there are two isomers. So we have to separate the two isomers, get the desired isomers. Say like S ibuprofen you take, then R again you racemize, again resolve. So big cycle of diastereoisomer formation, separation. So this we can avoid by using the asymmetric catalysis. So in asymmetric catalysis, we can directly get the single isomer. There is one university, I don't remember the name, but the company known as Chiral Quest in China, they have developed wonderful asymmetric catalysis methods, and they are producing single isomer. So immediately they become competent, cost competitive to us, because we produce both the isomers, then we form the diastereoisomers, we separate them, then again racemize, again separate. So we lose we build up the cost. So China has developed that and they have become cost competitive. So why don't we do that? So these topics should be introduced, and whatever drugs we are using in India, all which need a chiral drugs, go on developing the catalysis methods, hydrogenation methods, the oxidation methods. There's so many good methods are there, and I don't think that much more facility or nothing is big facilities are required for this. The colleges can take these small programs and then develop and offer the technology to the industry. They can get money. Means I see many of the non-grant colleges now finding difficult to run, but if they are devoting their time to technology development, they can earn a lot of money. They can run the college and they can earn profits if they devote their scientific knowledge to the needs of the industries. Then the process engineering also we are lagging behind, and I'm strongly against that mathematics should not be there at the UG syllabus or at the 12th level for pharmacy. See, mathematics means technology. Engineering is an applied branch of mathematics. So mathematics must be there, and you should, because pharmacy is a technology branch. It is not a medicinal branch. It's not a medicine. Because medicine is completely different, I would say. Many of our students, they feel that we are very near to hospitals or we are very near to doctors. That is wrong, I'll say. Because medicine is completely a practice-oriented course. It is totally different. We can give support to medicine, but we cannot be very close to the doctor. We are a technology people. We have a different, we develop a technology to deliver the drug properly. We have the novel drug delivery systems. So many things are there which we can do, but doctors, they don't bother about that. They have a different type of syllabus, different type of say, I would say, the profession, which is practice oriented. It is not directly compared with the pharmacy. Then another topic is there with the fixed bed hydrogenations. Still there are limitations in India, which is very cost competitive, which can reduce the cost. These technologies, when there is a company in Hungary, Thales Nano, which has developed the instrument which you can use in the lab. You can take the pressures up to 50-55 kg without using hydrogen. They have the systems to produce hydrogen from water. So the water can generate hydrogen, which is required, and you can carry out your hydrogenation reactions very simple and at very high pressure and very high temperature also, and get wonderful results. So this can give, this is at the research level. You can the college can have this instrument, and they can go further to offer the technologies to the industries. Now in the UG syllabus, I would say this already I discussed about the role of organic chemistry, biochemistry, and microbiology. I strongly feel that the first year and second year, we should not have semesters. Actually, the previous course, even Dr. Wakode also has studied in the yearly pattern. I'll tell you the difference. When you have the yearly course, then there is a very good concept development, because we don't learn at the B Farm or wherever we learn to develop the skills. We don't get this training to pass the exam. So this concept should be removed. See, the examination has got only 20% importance, and the skill development is 80%. You should gain that knowledge over the time. So 12 months. So 12 months, sorry, whatever, 8 to 10 months, whatever we have, except the holidays, your maximum time should be spent for hearing the lecture, interacting with the teacher, doing the practical. Complete attendance, say 90-95% attendance is important. Not just giving exam and passing with 60%, 75% is not important. So the first and second year, if we keep the yearly pattern, then a very good concept development means all the basics they know. To apply the basics for either pharmaceutics or ecology or cognosy, wherever you can apply, and the student can go to the molecular level. any chemical you are using, maybe in formulation you are using starch, then the student must get in this mind what is starch. Say like it is a mixture of amylose, amylopectin with this alpha glycoside. What is the structure? What are the groups involved? So whenever he is mixing that starch with his API, whether there are any interactions, he knows the API structure. If I compress it, because many times we observe that even ibuprofen forms ester with the starch. So these are means there are many interesting things. So when the student goes to the molecular level, then and then only he will understand. But if he doesn't know the structure of starch, then he feels that he has to take that and then make the granule and compress a tablet. He doesn't think beyond that. And there is no initiation of any research activity or any novel idea is suppressed. But if he understands the structure, even all the excipients, he should know the structures, even the lubricants, the colors. Why, when you see the structure of the color, there itself he can see that this color has no nitro groups. Say nitro groups are carcinogenic, the nitro compounds. You can see the metronidazole. You discontinue immediately when you get the therapeutic effect, because the prolonged use can cause cancer. So looking at the structure, he knows that what are the hazardous things here, or how it can undergo metabolism in the body, which enzyme can attack which group, how it can oxidize, what metabolites are formed, how it is excreted. So everything comes to his mind. So when you understand the structures. And now solubility, I can see there are many drugs which are very poorly soluble, and not good effort have been done to solubilize them, particularly the sulfur drugs. Very insoluble. So when he knows the structure, he know that which salt I can make, which derivative I can make without affecting the activity, how I can solubilize it. So a potential research idea can be clicked in his mind. Suppose he means provided he knows the structure. Similarly, antioxidants are there, how they act, what is the structure, what are the responsible groups for the antioxidants, then even the binders. So many things are there in this by which he can understand all these things. Now, there are many topics which are not, I think I don't find these topics in the current syllabus, like the stereochemistry, which is a very important part, I would say, very important part of the medicinal chemistry. And whenever, particularly when you are conducting, when you are at the PG level or when you are at the PhD level, when you are conducting a research, it's very important that how one isomer is active and how another isomer is inactive, that you understand only by its chiral structure. And in stereochemistry, even in the steroids, how the reactivity changes from alpha to beta, the 19 methyl group or these methyl groups in the steroids in the alpha position, the beta position, that is, he can understand better. So you must teach a very good stereochemistry to this in the first and second year, using the models, using the many of the advanced techniques. And again, I'm telling you, while teaching, I'll advise all the teachers that now I know that there is a corona situation and then we have to have this online teaching and all this. But whenever it was not there, or in future, hopefully it will go away. But whenever you go to the college, you only teach with the chalk and the board. Don't use PowerPoint presentations for teaching. I'll tell you the reason. When you start teaching, when you start some topic on a blackboard, you start at some point, and the student's mind starts, means he look at the point and then he goes further. The teacher starts writing a reaction or writing concept from there, and this concept is slowly developed in his mind, and then he, after 10-15 minutes, he completes that and he explains step by step, and then that concept is developed in the mind. But whenever you put a slide, the entire slide is open. The student cannot concentrate at that point where you started. He sees the entire thing, the first and the last. It is a flash, like it comes as a flash. And it cannot be memorized, it cannot be understood. you can use PPT only for the presenting the data. Suppose you have the spectra, you have some data, you have statistical, you have a bar graph, that is okay. But don't use this PowerPoint presentations for teaching. I traveled everywhere in the world to many institutes. I was a visiting professor, I went there. But all the professors, even today in US, Europe, China, they use chalk and board. They use PPT only for at the research level where you present some data. But when you teach, it's a different thing. When you present, your research paper is totally different. So teachers should use. That is what I feel. You can means it's it's not a binding, but I'll just sharing my ideas. Like now the physical chemistry also, there should be good knowledge of physical chemistry for the pharmaceutics, material science. We are very it's not, we have a limited access, means I think very poor knowledge to the students about the physical chemistry. So then they can do very good formulations if they know very good physical chemistry. Then synthetic chemistry, as I know that it's the only way to come to a cost effective routes of synthesis. And there are many, it's a very big topic. And now I come to the advanced analytical techniques, where I think they have removed, I think, from the currents. I don't know, but it is there or no, I don't know. But like mass, you can say, when you do the BA/BE studies, the bioequivalence, when you want to introduce a drug in India, which is approved in Europe, you must have this BA/BE. Now, there the metabolite studies, they are based on LCMS. You have to identify the metabolite only on the mass basis. You get this metabolite in this percentage identified by the mass detector, and then you confirm that this drug can be approved in India because it has got the same profile of metabolites. It has no other toxic metabolites or whatever. So this is understood only by mass. Now I'll tell you a very important part, like NMR, proton NMR, C13 NMR. You can see you any dossier or documents you submit either to USA or China, wherever in Europe. Now they recommend, they also are asking for C13 NMR also must be there for that particular drug or whatever formulation wherever used. And this knowledge must be there when you are doing the documentation. You must know that whether whatever document I'm submitting, whether they are correct. So unless and until you know NMR and C13, you cannot say proton NMR that the documents are correct, I'm submitting. They may get rejected if I don't have the knowledge. Now, another important part is the carbon, hydrogen, and nitrogen is a 3D NMR. I mean, these three NMRs, like carbon NMR, hydrogen NMR, and nitrogen NMR together can establish the structure of the polypeptides. It's complicated, but what is the great thing about it? I'll tell you. Because we do a lot of computer-aided drug design, the theoretical drug design, and in that case, maybe we consider the solid molecules and whatever the mechanical things. But whenever you try to conduct the activity for such compounds, and then you find a very different result. But if I have identified some enzyme where I want to block certain site, and I know that this drug is undergoing binding at a particular site, and this I want to prove, then the carbon, hydrogen, nitrogen NMR is very important, because you can establish the polypeptide structure using the carbon, hydrogen, nitrogen NMR, 3D NMR, and then you can interact your drug or test molecule with the enzyme, and where exactly it binds in solution, in water solution, you know by CHN, carbon, hydrogen, nitrogen, 3D NMR. So this gives a identification of the site where the molecules are binding or they are not binding, and giving the particular therapeutic response. So Japan has got a very big NMR park on this type of 3D NMR where carbon, hydrogen, nitrogen are used. And this NMR can also give that whether this amino acid is R or S, or this methyl is projected outside the helical structure or it is inside, so many things. So this becomes actually the future, that I can, instead of doing the molecular docking, on the computer and taking the docking scores or doing the homology modeling, you can, if you study this 3D NMR, then you can have the real docking picture, the enzyme where exactly the molecules are binding. Now, more lacunas are there in the PG syllabus, I would say, that the polymers are not taught to the PG or at UG levels. And now you can see that Udrogit, so many companies are using Udrogit and they are using it, it's a commercial product. There are many polymers, and we just publish so many papers using these polymers. But then we don't have our own Udrogit. We have to import Udrogit. So if I put a course, entire course, polymers in pharmaceuticals, there is a good Marshall Dekel series. There is one book, I don't remember the year, but the publication by Marshall Dekel series has got a very good book on polymers in pharmaceuticals, and which is very, very important. So there are so many areas, like particularly oncology, you have a lot of scope for a targeted drug delivery. Very, very large, because all the oncology, you know that all the drugs are very, very toxic. So if you have a targeted delivery, and there are many, many opportunities for this particular topic for having the novel drug delivery. You can see that in Taiwan, the people are using even fullerenes for the novel drug delivery. With the fullerene carrying the drug and then delivering at a particular site. So very, very advanced techniques are using. But if you know the knowledge of polymers, then you can do all these things. And in computer-aided drug design, this is also very good. Actually, I was also working on that. I have 3 PhDs on this topic. But here, I would suggest that the designed analogs, you must have, see that they are synthesized, and it's not a small number, but you can go for a large number of synthesis. Again, here the knowledge of polymer is important. If you use the Merrifield resin, or if you use a resin to protect some group, then you can have number of libraries. Actually, the library origin come from the Sofia, I think, the Bulgaria, with the Merrifield synthesis only, the compound libraries. So there again, you need the knowledge. But then computational drug design, very good area to work. But simultaneously, you must also see that you synthesize a library of compounds and then check that. Then chiral technology must be taught at the PG level. You must have a special elective for this, I would say. And it should be practice oriented. They should have the practicals. They should conduct some asymmetric hydrogenations. Say the small hydrogenators are available. Small now, as I told you, the Thales Nano Budapest is offering that instrument. The small catalytic hydrogenators are available. So if you make your students aware with these techniques, then there are very good research and job opportunities outside. Because I am associated with so many industries, and then when I conduct interviews, I have to screen for 200, 300 students, so candidates, for the better 10, 15 candidates. So that is what is the failure of our teaching system, that we are not able to give that much knowledge, or the university or the policy makers failure. So you should, if you have such techniques, then definitely I would say that we can produce very good skilled HR for this. Then antibiotics for fermentation technology must be introduced in the pharmacy at the PG level, definitely, so that one can, at least they can think that this is that. Maybe after 5, 10 years, but we'll get antibiotic, our own antibiotic. Now we are dependent. We have to import all these antibiotics. Then for PhD, I would say that there must be an industrial assignment. Without industrial assignment, no university should award a PhD. Whatever is the topic, I'm talking about only science and technology, not the other faculties. But unless and until he has completed certain industrial assignments, in China, the universities, they have for every PhD students, there are 5 industrial assignments. And these industrial assignments, just not some project and not get some certificate, they have to complete and commercially prove that this product was produced in so much quantity and this has saved this much, or it has lower effluent or whatever. So all these things they have seen, and then the PhD degree is awarded. So it is completely applied. One thing, one person is not doing only the work which is not fruitful. Whatever you do, I don't say that 100% fruitful, but at least 20-30% fruitful work if you do, that is sufficient. We can get a lot of things done with this. And now I would say that I'll request Mr. Sharad that I want to just get the feedbacks. I want to discuss with the audience.
Yeah, definitely, sir. Thank you very much. It was wonderful session, and now the session is open for the delegates. If they are having any suggestions or any query, sir would like to answer your queries. Really, sir, it was very wonderful thing that you have mentioned.
I don't know, means whether you feel it is a good, or whether it's not from academic, it is some like sort of policy type lecture.
Of an hour.
Sir, may I?
Dr. Sharma, please.
Good morning.
Good morning, sir.
Yes.
So your lecture, it was a full lifelong experience in industry as well as academia. So I am highly impressed with this. What you have talked is all based on your practical experiences. Now, I think most of the attendees are the young generation people. So I think it must have created a confusion in their mind where they should go back to the chalk and duster, or they should restrict to whiteboards, or they should adopt the latest technology. Because 2-3 things I also has been always in confusion in one of the universities which say we are world level following all this nonsense, all that. So they say only teach in English, not even a single word should be in Hindi. And when I see the lot of students going to such universities, I have to say 4 times in Hindi, then only they understand. Maybe because they have admitted 2, 3, 4 foreign students. So they complained if you say something in Hindi, So that has been always a challenge, whether I should make this subject to be understood well, or I should restrict to the foreign language, which 90% of my students are not understanding. Number one, 2-3 points, so you can together. Then second, I already told you, UGC, AICT, all regulatory authorities, they are pushing us more and more towards latest technological tools. But when teaching medicinal chemistry, or when teaching the mechanisms where you have to move the electrons with the help of arrows and all that, what I understood that it should be chalk and duster. Otherwise, a student doesn't get the insight of it. This is my opinion, but these days, even the authorities or the trends in general, they are pushing us more and more towards the technological aspects. And people are showing surveys also that this online has got this much success. And the third one is, I'm talking about 10, 12 years back when I was heading some institution. So I was talking to the top authorities in the university that most of the senior pharmacy people, they say it should be annual system for our students, as it is more of concept building, as you said, in the first and second years. So I talked to the dean of colleges there that, sir, for pharmacy, you kindly make it a yearly system. So he started laughing. Dr. Sharma, what are you talking? The world is going from annual to semester, semester to trimester, then monthly assessment, and then day-to-day assessment. Why you want to go back for pharmacy only? Because our university is handling hundreds of courses. So that made me to shut my mouth. So I'm really confused. So would you like to highlight on all these points?
Yes.
So thanks for your elaborative session, and these are some of the confusions. Okay.
All the three points I'll try to answer. See, like explaining the subject in their own language is always very good. I'll tell you, any book published in English anywhere in the world, is immediately converted to Chinese and circulated to all universities in China. So they have this system that they will understand in their own language. Even they have the computers, the CPU, the programs in Chinese. They can communicate in Chinese and they understand that. So it is no problem at all if you use the local language. Understanding, see, the understanding has no language, I will say. Either English or Hindi or whatever, understanding is understanding. Suppose he gets the concept and then he is able to reproduce, he should apply his mind, then no problem. You can use Hindi or local language, but then you must be capable of giving, teaching that part with the full details and then get the feedback also, whether he has understood or no. No problem at all. This is. I think the second point, what was the second point? About the semester.
Yeah.
So semester, so our college, I'll say, now I'll give the example of our college, Nasik College, was, since our Pune University was adopting only the yearly pattern, and we have proven well, like in the gate scores everywhere in the industry or in the wherever the admissions they get or wherever industries we have got very good response, and even USA or worldwide our students are there, and they are even at the vice president, the director or manager level. They have done very, very well because they had a very good concept, very good fundamental understanding of the subject. And this is possible not in the semesters. Why we follow the semester? Because US wants to do our students the semester. Why we work for US? We should see the development of our nation. Are we spending money for producing the HR for US? No. We need to build up our own industry. We don't put our syllabus that students should go to US. Why we should worry about US? And even if you want the semester system, see the first and second year you should not have, because semester they become just mechanical. You take the sessional exam, you take this. The teachers are completely busy only in exam and assessment. They don't know whether he understood or not. They are also mechanically writing. So first 2 years you can have, but next say third year, final year, you can have 4 semesters, because now he has a concept. He can apply that concept and can be used to the semester system, but not for the first and second year, because it's a very vast. Pharmacy is a very, very blend of so many things. So all these things they should understand. Then there must be a concept development. And I think the third point about the PowerPoint presentation and modern techniques. See, modern techniques are at that there whatever it is. But if you do not understand, means I found many of my PG students, they were downloading some ready-made PowerPoint presentations from the net, and they were going and then delivering some lectures on very, very different topics like biotechnology and all this. And now then I asked them, if you go and deliver there and somebody is asking you, explain this part, how you can answer this? Because they think that this PPT is available, I can do any subject I can deliver. It's not that. Who can give a PowerPoint presentation? He has a thorough knowledge of whatever slides he's writing there. So see there, whatever modern technology, it is there. I told you that even at the presentation of a research paper or your PhD Viva, it's a different level. But right now, from the first the 12th student passing and joining the college, and then you are showing PPT. What is the use? Wrong completely. You explain the concept on the board. It's not that India, US has that. They are using the chalk and board. Big universities where the professors have 100, 700 publications. They are going and with the chalk and then they are explaining. The UG students, they are not using PPT. They are using PPT for the only the presentations at the higher level, at PG, PhD level. Any other questions, Sharad?
Good morning, sir.
Yeah.
This is Dr. Gajanan from Rajesh Shah College of Pharmacy. First of all, regards, sir. It was a really nice session. पण आता मी तुम्ही जसं म्हणले सर की लँग्वेज बॅरियर नाहीच आहे, तर सरळच प्रश्न उचलतो सर मी की एक मराठीत म्हण आहे सर की मांजराच्या गळ्यात घंटा बांधायची कोणी. जसं तुम्ही सांगितलं की
गजानन सर.
हा सर.
गजानन सर, because we have lots of participants who do not understand Marathi, it
Okay.
would be better that we speak either in Hindi or in English.
ओके मैम। तो ऐसे एक मराठी में कहते हैं कि वो बिल्ली के गले में घंटी बांधेगा कौन। तो चीजें ऐसी है कि आई वाज फुल टाइम स्टूडेंट इन गवर्नमेंट कॉलेज ऑफ फार्मेसी अंडर द गाइडेंस ऑफ डॉक्टर मयूरा काले। बट ऐसा सिनेरियो है आज की लोग पार्ट टाइम पीएचडी करके डिस्टेंस एक फिनोमिना है कि डिस्टेंस पीएचडी होती है। मुझे पता नहीं था ये कॉन्सेप्ट ऐसा भी होता है, नहीं तो मैं भी पार्ट टाइम ही करता था। तो फुल टाइम आई स्पेंडेड 6 years इन गवर्नमेंट कॉलेज ऑफ फार्मेसी। बट लेकिन ऐसे कुछ लोग हैं अभी, हाँ औरंगाबाद अंडर डॉक्टर मौर्य सर का स्टूडेंट हूँ सर मैं। तो पार्ट टाइम, हाँ सर, तो ये पार्ट टाइम का फिनोमिना कहाँ से आया? तो ये बात ऐसी होती है कि गले में घंटी बांधेगा कौन। तो बात ये है कि आप जैसे बड़े लोग हैं सर, तो हम अभी तो फ्रेशर है सर चीजें करने के लिए। तो आपकी ओर से कुछ राह मिल जाए तो डेफिनेटली हम कुछ कर पाएंगे इस चीज में कि ऐसी चीजें होनी नहीं चाहिए, या फिर जैसे आपने बताया कि इंडस्ट्री के ओरिएंटल पीएचडी होनी चाहिए। तो इन माय पीएचडी आई हैव सिंथेसाइज्ड अराउंड 40 डेरिवेटिव्स सर, लेकिन उसका कोई कमर्शियल यूज है नहीं आज। तो आपने जो बोला सर, इट इज रियली आई ओपनिंग फॉर मी। तो अगर आपकी ओर से कुछ मिल जाए डेफिनेटली, तो आई विल इम्प्रोवाइज माई सेल्फ टुवर्ड्स माई स्टूडेंट्स।
See now, see like everybody has what they think that at MPharm or PhD they should discover. Now, why I would say that the discovery of a drug is not a small game. It's a very big game, and it takes a lot of money and lot of time. So like almost 15 years, 20 years, big programs, $800 million, $1,000, $2,000 million, big game. So we are not there. It's not our concept that we just work and discover some drug. It's not that. So what I feel that even if I say simple, now I'm conducting a project at the sugar factory for producing paracetamol. Now paracetamol is a very small simple molecule, but India, the paraminophenol is importing only from China and they're making paracetamol. So now just if I make a process from phenol to paracetamol, or what a simple process, that is a great achievement or the great help to the nation. It's not that you go and then discover something. So my feeling is that such industrial concepts of the projects, whatever, then many areas are there. Even they successfully produce some technology, then they should award the PhD, because that is the real commercial. That the nation has stopped the importing, we save the foreign exchange, we can spend that money in India for the development. Otherwise, we have to earn and then give it to the oil import or for the intermediate import, we have to put all that money. So even the technology, simple technology development, or even the NDDS, it's a big field of novel drug delivery system. Simple. There should not be very novel things, but it has helped the industry, it has helped the patient to get cured properly without pains. That is also achievement. Why one should worry, and then why we should stress that we should have the publication in the particular journal and all this. You see the commercial success. Here, what we try to do, we try to do something that it will be suitable for a particular publication. Then we work for that. And what is the output? There is no output. The paper is there for 50 years, 100 years, it is like that there. See, I'll tell you my example. I have just, actually I am from a chemistry, farm chemistry, but I worked on a formulation project for burn cure. Then this silver sulfacetamide, the absorption is very poor. Okay, it used to take a longer time for the burn care. Then what I did, I used some silver salts of fatty acids and very little silver nanoparticles. So it has cut down the curing period, the total healing period, from 40 days to almost 12 days. So in 12 days, it is completely cured. So this is also achievement. So we can file a patent on this, Indian patent, and that can be credit as a like publication. You can have a patent in it, you can get a patent granted, and you can, this will be a credit for his PhD. So we should work, I think, in this direction for our this thing, and then need not to go for a distance PhD and all this. If you have, see, like you must be mentally prepared that I want to work in my college. Then everything you will get. If you say that I don't have this, I don't have this, if you work in your college, you will automatically get. And there are many institutes, they will help you for getting the spectra, all these things. But it is the basic, your determination. I want to work. This is important.
Thank you, sir. Thank you for inspiring.
Yes, sir. Good noon, sir. I am Prabhu Sharma from Delhi Pharmaceutical Sciences and Research University. And
Excuse me, sir. Excuse me, Prabodh, sir. I request someone has put on the microphone. Please mute your mic. All of you mute your mic. Only the person who's going to speak should put on the mic. Prabodh, sir, please.
yeah, good noon, sir. I am from Delhi Pharmaceutical Sciences and Research University, and you have presented a very wonderful talk, so appreciation for that. But I have a common question that every synthetic medicinal chemist in academia is facing. Now, the job of a medicinal chemist or a pharmaceutical chemistry teacher has, in research, has confined right from synthesis of some small heterocyclic molecules, some molecular modeling or docking studies, biological evaluation, and publishing a paper. That is what most of the people in pharmaceutical chemistry are doing in our country. And there is a great link missing, first application part, and that application seems far away. So much money is required, manpower and expertise. So, as an expert, how do you suggest that? How can a medicinal chemistry can go ahead in that direction?
Yeah, actually, if I want to really work in this direction, then there are say many companies in the world to whom you can submit your virtual libraries first. Means for a particular lead, you develop a virtual library first, and this virtual library, if you submit them, then they can select. They have some software, I think known as PASS, which has a capability to identify that this structure has got which type of possible activity, and it gives some score also, that this is this score, and they will give that feedback to you, and then you know that probably I should select this area or whatever. I mean, you have the scale there, and then only that part. So suppose you design a 5,000 screening virtual library, then you have selected some 40, 50 molecules, and then you can synthesize only those. And even I would say that if you get the 90% purity also, you get some activity out of that, and then you want to also get supported by some funds out of that. Then if you submit these samples to that company, then they hold with them, and then wherever their website where they are displayed all the structures are referred by the world best pharma companies or discovery companies, and then they buy that on a milligram basis, some few dollars for a milligram, and then you also get funded for that. Means the institute can get some money from that. So I think there also your purpose is served, that you have done something, say towards medicinal chemistry. You have done synthesis, you have done some activity, and you have some logic, some little logic that this can work. Otherwise, means I think making virtual libraries and getting assessed is the best method for this type of work.
Okay, sir.
I'll share you the details later on to Sharad Wakode. Then he can give you. You can contact.
Please, if you can have the details of those companies, that would be a great help to us. Thank you, sir.
I'll give.
Prabodh is associate professor in our DIPSAR University.
Sir, mic is not clear, sir.
Dr. Prabodh is associated with DIPSAR University only as an associate professor.
Yeah.
He was earlier with the Kurukshetra University.
Okay. So definitely, I'll help you in this. And
Prabodh has very good publications in high impact factor journals, sir.
Yeah. Okay. Very good.
Thank you.
I know DIPSAR. I know, with I'm coming there from once. I think I have delivered a lecture on some spectroscopy or somewhere. I don't remember with the year, but I was there to your institute. You are a very good institute, and I think your that animal house is very beautiful. I've seen that, the rabbits with the burrows and all this. I've seen the institute. I have good regards for this.
Natural habitat was there.
Any other questions? Any other questions
Hello.
from the delegates?
Hello.
Yeah, please.
Yes,
Sir.
Sandesh.
Yes, sir. Good afternoon, Sandesh Luda from
Yes, yes.
Maliba Pharmacy College.
Yeah,
Your student, sir. Very good insights. But
Put on the
what?
video, Sandesh.
Yeah, I'll try, madam. Hello. Yeah. Sir, there is
I'll
a
tell
simple.
you. Go ahead. Go ahead.
There is a simple issue which we face is lack of interest from schooling itself. The MPharm chemistry courses in India, around Western India, I have almost gone through many colleges where they don't have admissions at all. MPharm chemistry, no admissions. Very few colleges are getting students who are attracted towards medicinal chemistry, and they have this fear for chemistry.
Okay.
So I believe that the education system, schooling, there has to be a change to bring the interest back in the students for chemistry. What is your stake, sir, on that?
Okay. See, like I'll tell you, if you see the jobs offered in the industry, hello?
Yes, sir.
Yeah. If you see the jobs offered in the industry, I'll give you an example of Nasik, okay? Glenmark, the general manager is MPharm chemistry, okay?
Yeah.
Glenmark Research Center, general manager is MPharm chemistry. GSK, the site head, MPharm chemistry, okay? Mylan, the site head director, MPharm chemistry. So the chemistry people have got a wide scope in the pharmaceutical industry rather than Sutix, I'll tell you. Because of the molecular understanding.
Yeah.
They go to the molecular level. So you must first explain this all your students, whatever. If you know the chemistry better, definitely you have got a good opportunity in the pharma industry or any other industry. Rather than Sutix or Coology or Cognosy, the chemistry has got upper hand. I'll see many of my students have gone to the higher level, the vice president. Even one of our students is a director in Sanofi at Boston, who is a chemistry. So many are there. I can give hundreds of examples. So it's not from the school. It's from the first year of B.Pharm. You must explain this. It is not from the school. See, the school is 12th science. Okay, these all general. It's not special. So when the students get admitted, then you must first, what is the scope? What you are going to do? I think the principal should address this to the students. When they get admitted, what is pharmacy? Then what are the scope? What are our past students are doing? What is the pharma industry? All these things. What are the areas you can work? Where you can do? What are the departments and the pharma? All these things must be explained properly with a very simple language, not giving very high fundas. Just give a very simple lecture, simple in their only their the original language, no problem. But they must understand that. Then this interest will be developed.
And here I would like to add to sir what he said. For kindling interest, one must have the practical hand. What we can do to the student is, see, we are lucky in chemistry. You mix this to mix that, you get beautiful colors. You mix this to mix that, you get effervescence. You mix this to mix that, you have so many reactions which are eye-catching reactions. If you show one of them to the students, they would be attracted to that. And second thing, what I feel is the attraction towards chemistry is reduced out of fear. Fear of the molecules, fear of the reactions, fear of the memorizing all that. So a teacher has a role in developing understanding of the student towards chemistry rather than mugging up. Most of the students, they are mugging up, and mugging up can never resolve the issue. It just drains out after the exam. So if they develop the interest and understand how the reactions are proceeding, what I observe is in medicinal chemistry, when you teach synthesis, we never teach them, how the synthesis should be developed. We teach them, add this to this is the result. It should not be like that. It should be, if OH is there, then this OH can generate so many derivatives. That should be the approach. And then you should give them further. This is the molecule you have to synthesize. And when this molecule has to be synthesized from this raw material, what would be the procedure you would adopt? You have to introduce this group here, that group there, that group there. So if they come out with their own synthetic route, naturally they are going to have a lot of interest. And salute to Sanjay Bagh sir, because you know, application is the most important thing, and here I salute engineers for that, because most of the projects they do, they have application. While in our pharmacy, 95% of the research is just the drain of the energy, drain of the resources without any outcome. I can always say, you know, in cognosy, what we do, we choose a plant, we extract. Extract has many components. Sometimes the component is separated, sometimes whole of the extract is used. So the outcome is nil. It is in the PhD thesis or PG thesis. The PhD is awarded and it is gone. So you have said very well, sir, touched upon such points which will, if implemented, India can really go a long way.
Thank you.
Wonderful presentation and the thought-provoking session for all the teachers. In the beginning, you said that you are out of the place. No, sir, you are most in place lecture, you know. It was most in place lecture, wonderful thought-provoking appearance.
Very good platform where all the budding teachers, so many teachers from all over India are there, and they are in the stage of their development of their career. So they can take advantage of this and start thinking in that point of view. Even Madam Parle is teaching organic chemistry, and
very good
with the gist and with such an interest.
I understood. She has I understood very well, and she has been so wonderful.
She teaches very nicely. All the
and Sharad, I need this recording if I can get this.
Yeah, we'll definitely.
Hello.
Available on YouTube.
Yeah.
Right now, after this lecture, if you go to the YouTube link, you can download from there.
Yeah, thank you.
Yeah. Yes. I can see here Dr. S.B. Bari, principal.
Yeah, yes, Bari was also asking. Bari was also asking for a link this morning.
Yeah,
he's there. So actually, I was exchanging some thoughts with Dr. Bari about the syllabus framing, and which I felt very bad when I found this new syllabus. Yeah. So how are you, Dr. Bari?
Fine, sir. Your lecture was very informative and thought-provoking. It was eye-opener for all chemistry teachers also and academicians also. Yeah. But you're told that is blend of academic and industry should be. It is a need of our. But how it can be proceeded with the direction, that is very major problem. Everybody understands, sir. Yeah. There should be the blend of academics and industry.
There's only one thing, Dr. Bari. That is, you should have an initiative. If you have in mind that I want to do this, then you can see there are several options. You can go to any industry, any further. Nobody will say no to you. If you say that, even you give me any small assignment, like just method development, it's okay. You start with a small thing. We can achieve big things.
Yes, sir. So we need your help also, sir.
Yeah, definitely. I'll also help you, sir.
Rather than help, we need his guidance and pushing force from behind.
Yeah. No, always helpful. That is no problem. I've been in contact since 10-15 years back.
Yes.
And you have rightly said, Dr. Bari. What happens is, see, PG student comes to you. You have hardly 6 to 8 months, truly speaking. In that, he has to complete the research and go. So most of the times, the projects allocated are whatever is feasible rather than ideal.
Yes.
So that is where the problem starts, because contacting the industry, sending, we do. It is not that we don't do, but Then industry says you cannot publish, you cannot use the data.
Yes.
There are so many,
No,
restraints.
but there can be way outs for that. I can suggest, so you will contact me. I can suggest that how we can do that. Definitely.
Yes, sir. And another thing is, so excuse me.
Yeah.
So since we are working in the private organization, so their focus is only admission.
No, but see, admissions, when you can attract the students, suppose placement of your college is very high,
Yes, yes.
then you can attract the admissions. So
Sure,
if
sure.
you associate with industry, definitely your placement will promote. Definitely the industry will respect your students that
Yes.
you can send.
Thank you, sir. Thank you.
Thank you.
Any other questions, please? Any other question from any participant? Wag sir, I'm just reading out some of the reactions. Sir, it is very nice interactive session. It is an honor to listen to sir and his thought-provoking insights. This is by Sandesh Lodha. Then let me read out some more. An awesome overview. Professor S.B. Wag has always been radical and utmostly practical. A blend of academia and industry is always the need of the hour. High time to see the change. Inspiring talk. And so much more, sir. Similar comments are there. So you can feel that how nice your lecture was.
Thank you.
Ramakant Joshi mentions that, sir, your lecture is very thoughtful. This is your out-of-box approach will definitely give you a very good career imparting in fulfilling the pharmacy future. So it will be definitely very good thought-provoking thing out of box what he is mentioning, definitely. And thank you very much for accepting our request. I know that sir was very busy with his assignments, but I requested him and pursued that he should be there in our quality improvement program, and he accepted. Thank you very much, sir.
Professor Sharad Vakode, I request you to present him a virtual certificate.
Yeah, it is my honor.
Yeah. Thank you.
Thank you, sir. Thank you. Please.
Thank you all.
Sir, we are going to present you a virtual bouquet as well of the flowers. Kindly wait for a minute. Sir, kindly accept this bouquet from DIPSAR.
Yes. Thank you. Thank you.
Thank you, sir. Thank you for sparing your valuable time and giving us in-depth insights how the teacher should modify their approach towards the practical aspects. Thank you, sir.
Thank you. Thank you all.
So here we end with the morning session of the 5th day, penultimate day of our quality improvement program. You all can proceed for a simple lunch because you should remain awake in the afternoon session as well and come back at 1:50. It is always nice to have you all with us. Thank you so much.
Thank you. Thank you, all the delegates.
Thank you, sir.